This meeting is being recorded.
All right, hello everyone, and thank you so much for joining us for the February Cell Modeling Seminar from the Center for Reproducible Biomedical Modeling. Today, it's my pleasure to introduce Dr. Barbara Bakker, who is a professor of medical systems biology at the University of Groningen and a Rosalind Franklin Fellow. Dr. Bakker has expertise in systems biology studies of metabolic networks and metabolic disease, dynamic network modeling, and enzyme kinetics, among other topics. And she has used experimental and mathematically driven computational approaches in her research to try to rationally identify possible drug targets in metabolic networks from multiple host systems. Today, she's going to explain how her group has used personalized simulations in a dynamic fatty acid metabolic model to better understand differences in disease severity between patients with metabolic disease, which shows a really clear application of modeling within the biomedical community. So welcome, Dr. Bakker.
Thank you so much. Thank you so much for this great invitation. I really think it's a fantastic initiative that you have this, and I have been following some of your seminars on YouTube because at the moment I could not be present, but I of course wanted to see what's happening before giving my own seminar. So let's start straight away. I'll start sharing my screen now. So this is what I'm going to share. Slideshow. So I think you can see my slide now. Is that correct? Yes, I see people nodding, so that should be okay. So what I will do today is discuss our long-going effort by now on modeling fatty acid metabolism in relation to disease and eventually in relation to personalized medicine for metabolic disease. So in our group and in the broader sense in our department, we are very much interested in the interplay between lipids and glucose or carbohydrates in more general in disease. So what's very well known to many people is that there is a strong relationship between obesity and type 2 diabetes. So obesity being an accumulation of lipids and type 2 diabetes being a disorder in carbohydrate glucose metabolism. This is a well-known disease. What's less well-known maybe are the inherited diseases, often monogenetic, like inherited disorders in fatty acid oxidation, and they have a severe risk of hypoglycemia, so low life-threatening blood sugar levels. On the other hand, starting from the carbohydrates, I have also colleagues working on glycogen storage diseases where there is a mutation in enzymes involved in glycogen metabolism. And those patients do not only have a disturbance in carbohydrate metabolism, but they, for instance, can also develop a fatty liver. So whereas obesity type 2 diabetes is, of course, occurring much more frequently, it's also quite complex and multifactorial. So may be surprising, but I'll show you why. We decided from a systems biology perspective to first start from understanding these inherited diseases because they are severe phenotypes. They have a system-wide impact, and they're also treated through the system, like dietary treatments are typically system treatments. So they give us a lot of insight in how this interplay between lipids and carbohydrates work. At the same time, we hope to give back something to these children and their parents with very severe diseases. And on the other side, we also hope to learn more about the system where much more prevalent diseases occur. So that's the idea. I will focus today completely on fatty acid oxidation disorders, but in the lab we also started working on type 2 diabetes now. So systems medicine, how do we see this in our group? What you would like to have is a personalized prognosis for a patient. So we are screening many children for diseases. So some come out as a mutant because there is a metabolic aberration, you find a mutation. Some of these mutants are really patients. Some of these patients run severe risks of, for instance, hypoglycemia, but some of them have very modest symptoms, so they may not run such a risk, so they may be overtreated at the moment. So who is who? And then if you want to treat them, which diet should you give them and which medication works best for whom? So the idea is that even if you have here in red, you can see my cursor as a pointer, I hope. If you have here in red a mutation, and you have another person with the same mutation, the surrounding network may be quite different. So here you see certain parts of the network are underexpressed compared to another person. Some parts may be overexpressed, and these may represent compensatory pathways or pathways that make things worse for the patient. So those differences we have to take into account when we want to understand the phenotype of even a monogenetic disease. So what we do in our group is we make computational models of these systems, and I will focus today on the beta oxidation of fatty acids, and we start from a very reductionist perspective. So reductionist in the sense that we think that the properties of the complete network should be understandable in terms of all these small interactions between the different components. So if we start from a human body with all its metabolites or a cell type or even a pathway, then we have many components. As biochemists, and I am a biochemist, we tend to take these apart and study their properties, study in vitro, in a test tube, how they interact. But this is often where reductionism stops. But true reductionism, of course, should be put to a test. So you should, from the properties of these components, you should start building up the network again. And you can do this in an experiment in synthetic biology. We do this in the computer. So in silico, from all the knowledge that we have about the components, we start building up a model really bottom up. And then, of course, you would like to see if this in silico image, if it resembles the original. If not, there is something to learn. And we have to go back to the lab, ask the right questions to get this right. If it's good enough, at least for the questions to be addressed, we can start putting in patient-specific information, like protein levels that may be different between different people, mutations, and then we can start seeing how a patient differs from a healthy person, but also what all kinds of molecular interventions would do on the system. So that's the philosophy of what I'm going to show you today. And as I said, we are very much interested in this interplay between sugars and lipids, and an important organ there is the liver. And if we talk about these children with their fatty acid oxidation disorders, they tend to have a problem in the fasting state. So these are young children, so the parents are very happy when they start sleeping throughout the night. And then throughout the night, the liver has to make sure that enough glucose is being delivered to the blood for all the organs, because the child doesn't get any food at that moment. So where does it come from? It comes, of course, from glycogen stores, but it can also be produced de novo. So once the stores are getting depleted, it becomes important that, for instance, amino acids are taken as substrates, converted to glucose. But this requires energy in the form of ATP. And then we need the fatty acid oxidation. So if there is a defect in the oxidation of fatty acids, then you have an immediate risk of low glucose levels. So this is a life-threatening disorder in principle. So just to get a little bit of biochemistry, I realized that most of these talks have been more on methods. I'm more a biochemist with specific aims and using modeling as a tool. So I hope to give you a bit of both worlds. But to get to the heart of this talk, it is important that you realize that lipid molecules and fatty acids in particular, they are long carbon chains. So if you have, for instance, an activated fatty acids of 16 carbons, we call it palmitoyl-CoA, then you see these 16 carbons here, and at the end it is activated by binding to a so-called coenzyme A, and you see it here via sulfur bond bound. And if this is oxidized, then it's broken between the second and the third carbon atom, which means that you're left with two pieces. One is a piece of 2 carbon atoms and the other is a piece of 14 carbon atoms. And the piece of 2 is also linked to this coenzyme A. So this coenzyme A is an important player linking many things in the cell. So from networking perspective, this is really our hub. And the C14 acyl CoA, it has very similar properties as the C16, so it can be broken down to C12, C10, etc. So that's the type of problem we are facing. If you then look at what kind of system we need to model, it's a bit more complex than that, because we have here the mitochondria. They are the energy factories in our cells. So these lipids, they are taken up into the mitochondria. They cannot be taken up straight away, so they're first converted into a carnitine ester, and these carnitine esters are being taken up, and then they're converted back to this ester with coenzyme A. And then only the story begins, because I just said we have this breakdown from C16 to C14, etc. But this is a set of 4 reactions. So if you go through one breakdown step, you go through this cycle of 4 reactions. And you have to go through it multiple times, over and over again, until you have broken down your fatty acid chain completely to acetyl-CoA, as we call it, so the C2 compounds. And those can be broken down further and generate more energy. So this is the type of pathway that we need to model. And I'm going into a bit of depth here, because you may realize that such a system is going to have special properties. First of all, what I like to point out is that we know inherited errors of metabolism associated with almost all of the enzymes. So a predominant one that we are focusing on a lot is this one, the medium chain acyl-CoA dehydrogenase, and that will be central in my talk. Another one which I will be talking about is if the electrons that are generated in this reaction, they go to an acceptor, a molecule called FAD, and if that FAD is not properly regenerated, then all these reactions are in trouble. But there are also inherited disorders, for instance, with this uptake system or other enzymes in the network. So what happens, and why do I think this is interesting for systems biology and modeling? Well, this MCAT deficiency, so this enzyme, it occurs in 1 in 8,000 people in the Netherlands, and differs a bit regionally. In many countries, it's in the newborn screening. These children are born apparently healthy, so the system has backup options. But if these children start fasting and then have an additional trigger, then they have a high risk of severe drop in blood glucose levels and sudden infant death. It's not just when they fast, it's with an additional trigger. So the system really has to be pushed hard to break down. The treatment is simple, so if you get them from the newborn screening and if you have them early enough, a regular intake of food and close surveillance under disease conditions like fever is usually sufficient. But somehow their metabolism is not robust. It can function most of the time, but a trigger can break it down. And we want to understand this robustness. And I think this is a systems biology question. But when we were looking at the system, we realized that it has very special properties. So if you look at these enzymes, so these are now schematically the enzymes, they're utilizing all these different fatty acids with different carbon chain lengths. And what you will appreciate is that we have multiple reactions, multiple substrates, but much less enzymes. At most, we have 4 enzymes doing the same reaction with a certain specificity for longer chains or for shorter chains. So in all cases, the metabolites are competing for the enzyme. So they are, you imagine they need a spot on the enzyme, and if their competitor is sitting there, they cannot be converted. So they have to share that capacity. There is also a positive side on this, because these enzymes have they're not so specific. So if one of the enzymes, like this MCAT enzyme, if it is deficient, then these medium chain substrates, they can also go to the other enzymes. So C8 can go to the longer chains, and C6 can, for instance, go to the shorter chain enzyme instead. So there is also rescue in this system. So we were wondering whether this competition now between the substrates for one enzyme, the fact that they are not just converting one substrate but multiple, whether this has to do anything with the robustness of the pathway. So we have a couple of research questions. First of all, is this metabolite competition, is this now affecting the robustness of the pathway? And later on I'll come to, if this is the case, and I'll show you, yes, it is the case, is this now physiologically relevant for the phenotype as we see it in our patients? And what does the surrounding network do here? Karen van Eunen, a postdoc in our group, she built a computational model. And as we have discussed now that these enzymes all do multiple reactions with multiple substrates, even though you see only few enzymes, there are many more reactions there. So we have 56 rate equations here, giving us 200 kinetic parameters, and they were all taken from the literature, from in vitro studies. Some of them we had to identify ourselves. So this is completely bottom up. We just put together in a model what we have and see how the system starts behaving. And the model is an ordinary differential equation model. So you see differential equations for each of the intermediate metabolites. So there are balance equations. So you have a velocity of production and consumption of each of the metabolites. And in some cases, there are multiple enzymes producing something, multiple enzymes consuming it. So then the balance equations become a bit more extended. And each of these velocities then depend on the concentration of a specific enzyme, but also on a vector called X, which contains the metabolite concentrations, which act on all these enzymes as substrates, as products, but also maybe as modulators. So this is the complexity that we're facing. And we built in this competition in the following way. So this is basic, very basic enzyme kinetics, in fact, but it looks quite complicated maybe. So these are velocity equations, and they depend on the maximum turnover rate of an enzyme, the enzyme concentration. The more enzyme there is, the faster a reaction will proceed. It also depends on affinity constants for the metabolites that are being converted. You have a forward rate, but the net rate also depends on the reverse rate. So there is a substrate being consumed, there is products being converted back. So the net rate is dependent on forward and reverse. That's all kind of basic enzyme kinetics, but what's special, of course, is that you have these competitors. So here we have C16 being converted, and then the other chain lengths, we're looking at this enzyme, so 16 goes in, but also C14 and C12 can bind and be converted. So for this reaction, they work as a competitive inhibitor. And competitive inhibitors and enzyme kinetics are put in here in the denominator with their affinity constants, the Kms. So this is how they start inhibiting each other. You divide by it so the rate becomes lower. So this is how they have an effect on each other. So Karen, as I said, started modeling this, and we wanted to validate this model experimentally. So what we can easily measure are these acyl-carnitine esters. So you have the palmitoyl-CoA coming in, it's becoming an acyl-carnitine ester, then you have all these conversions inside, but all these different chain lengths intermediates, they can also be converted back to the carnitine and exported. And those are also exported to the blood, for instance. So in clinical diagnostics, this is used a lot. So we can easily measure this. So it would be a nice simulation for experimental validation. So Karen started looking, what if I give this C16 coenzyme A? The first thing you see appearing then is C16 carnitine ester. So here we have the carnitine ester. So you see it appearing, but then it goes into the pathway, so it's disappearing as well. It's a relatively long time scale. And then you see all the different chain lengths coming up and going down. And especially a shorter one, C6, tends to tail a bit longer and a bit higher. And this was the experiment. She isolated mitochondria from rat liver, from where we had also most of our parameters. And this is what she saw without any fitting. This is really bottom up. So she saw the C16 coming up and going down, really at the same concentration scale, same time scale. And she also saw the C6 really going higher than the others, and then many of them going up and down like at much lower concentrations. So this was pretty reassuring. Then we started using the model for some predictions. And these are steady state solutions now. And here you see the flux as a function of substrate concentration. And increasing a substrate has a physiological meaning for us. It's what happens during fasting when we start utilizing our resources from the adipose, so that also happens in our patients. It's also what happens in obesity. People have higher fatty acids in the blood, so higher substrate for the liver. And this is what we observe. We see, as we would normally expect, more substrate, higher consumption flux, but then we see a very sudden breakdown, and that's not what you typically see in biochemistry. In biochemistry, you expect to see saturation, and we didn't see this. So this is clearly a property that comes from this model, and that is surprising. Well, we could start to understand a little bit in the model what's happening there. What we see is that this specific coenzyme, the coenzyme A, tends to decline. First a little bit, but when this breakdown happens, you see it declines very steeply. That's the black line. And all these intermediates in the pathway, they are esters linked to coenzyme A, and you see they start accumulating. So what effectively we see is a huge accumulation of metabolites inside this system, and that's when it breaks down. So we wondered then, does this have anything to do with this promiscuity of our enzymes, the fact that they use multiple enzymes that are, multiple substrates that are competing? So we did a thought experiment, an in silico experiment, just to see what would happen if this competition did not exist. We know it exists, but what if? Does it have any consequences? So in silico, we could partition each of our enzyme pools in small portions, each of these portions having a dedicated task. So that means that if you now look at our velocity equation, you take just a fraction of the enzyme for that specific task. So now suddenly we take just a number between 0 and 1 for that specific task, and all the fractions of course add up to 1. So that would lower the rate, but at the same time we take out the competition factor, because we now do as if this fraction of the enzyme sees only one substrate and another fraction sees the other substrate. So this is an effect that decreases the rate, but removing the competition would increase. So in principle, these things could cancel out. So what would happen now? This is what we saw. So this was the original, this is the real biochemistry. And then if we remove this competition artificially, you see a very nice saturation curve. So at low concentrations, it doesn't matter, showing that in principle, the two things can cancel out. But here at high concentrations, you see that apparently this competition was the cause of the breakdown. And if you also look at the free coenzyme A, that was declining originally, and in the new model without the competition is not. So clearly there is a relation between this, the way the system is wired and this risk of severe breakdown. So Anna-Claire Martinez in the lab, a PhD student, she analyzed this model, and I won't go into much detail, but I think for the theoreticians among you, it's an interesting paper, because she used a combination of metabolic control analysis, and she used a theory analyzing the regulatory strength in this network. And she used this to identify a vicious cycle. And I'll just show you what is in the end the summary of what she found. So if we increase the substrate for this pathway, what happens? Well, initially, this is the rate limiting step, the first uptake step. It's really having all flux control. When you give more substrate, it becomes saturated, it gets less control because it's activated, so something else becomes limiting. And that is this enzyme. So if you go through around, it's the last enzyme of the cycle. And then you see when it becomes limiting, its substrate starts accumulating a little bit. That's no problem in itself. But the previous enzyme has an equilibrium constant of 10 to the minus 4, which means that at very low product concentrations, this already is inhibited and tends to equilibrate and cannot proceed anymore. So if this starts accumulating, this also starts accumulating to very high levels, and then still the pathway proceeds, but it is clogging up already here. Well, then you go to the beginning of the cycle. There you have the acyl CoA, but it's the beginning of one cycle. It's also the end of another cycle. So as the end of a cycle, it would inhibit this thiolase enzyme because it is product. So it's here you see the product inhibition. So this enzyme that was already getting some control on the flux, becoming limiting, is now even further inhibited. If we get this clogging up of metabolites, we are sequestering this coenzyme A, and it's one of the substrates for this last reaction to produce the C2 fragment with CoA. So that goes down. So it's even further inhibiting our enzyme. So this is how you get a vicious cycle, why the whole thing at some point can break down. So this is a serious risk that is inherent in fatty acid oxidation. The question is, of course, why don't we see it so often in reality? Why are we all healthy? But inherently, if you just take the naked biochemistry of only this system, this is a severe risk. And Marcel Viaralara, and at that time still a master's student, now a PhD student in the lab, he also looked what would happen if you would just activate this enzyme. And it is a regulated enzyme in the cell, so it can be activated. And what you see is if you have the original model, then you see this clogging of CoA esters, high concentrations, the breakdown of the flux. If you activate this enzyme in blue, you see no accumulation and you see a nice saturation curve. So this is, I think, quite an interesting cue where we can really manipulate the system with something that is targetable and also in vivo endogenously regulated. Also found another interesting property of this system that was by Fantau Abagas. He's a mathematician, postdoc in the group. He found that there is bistability. So this breakdown can happen, but if you go in one direction, it happens much later. And if you then go back, you can stay for much longer time on the lower arm. So there is a region, and depending on the parameters, the surrounding network, this region can be smaller or bigger, but there is a region where there are two possible steady states. And depending from where you come, you will be in what you might call the healthy state with a good energy flux, and in the disease state with a low energy flux. So if you happen to be as a patient on this branch, you might need only a transient perturbation to get on the right track again. So I think from a medical perspective, this is also interesting that transient perturbations could cure within a certain window. So that's the very basic properties of our network. You would wonder, of course, is this now physiologically relevant for our patients? And I must say we have to be very modest, but we started to do modeling of our patients. So we started looking at these acyl-carnitine esters again, because they are measured in the blood, so they can be used as a readout, and we have loads of data from the clinic of patients' acyl-carnitine levels. We started doing this for the same disease, MCAT, but what you then see, it's a problem of the medium chain. So it is this enzyme, which is converting C4 to C12. Its best activity is for C6, C8, C10. So if this enzyme is deficient, it's no surprise that we see its substrates accumulating. And that's what you see in patients. That's also what we see in our model. And it doesn't matter whether we take the realistic model, the competition model, or whether we take the artificial model. We also see it accumulating, maybe a bit lesser, but still it accumulates. So that's no surprise. You wouldn't need to be a modeler to get insight into this. Where it becomes interesting is when we start looking at this regeneration of this coenzyme FAD, so the electron acceptor. Because if this is problematic, you get a problem in all the different reactions at that level. So the first step of each round would be problematic for each chain length. It's very rare the symptoms range between very mild, when it's a partial deficiency, or very, very severe, when it's a larger degree of deficiency. And you can imagine that it's severe because then none of the chain lengths would be converted, so the complete fatty acid oxidation is down then. I was very much triggered when I saw patient data, because what I found then was that all these patients, they have an accumulation of acyl carnitines, and you see the patients in blue here, for all chain lengths, so C4 to C16. And here it's even more extreme, so the concentrations are higher, the controls are the same, but they are almost invisible because there is such a high accumulation of especially the C4 and C16, but also in the middle they accumulate. So Karen, I would not have expected this in the first place, because if you are a C12 somewhere in the middle, both the production and the consumption are inhibited by this lack of regeneration. So I thought this would balance out in the end and it would not give an accumulation of anything. I was quite surprised. So we started modeling to see if our intuition was right here. And Karen modeled this just by changing the degree of reduction of this cofactor. So that way she could simulate the two different patients, which are in fact characteristics of the extremes that we see in our population. And here she had the normal model, the computational model as it should be. In red the controls and in blue the patients, and you see they show pretty well the same pattern, and also they could show the extreme case, a more extreme patient with a very high C4 accumulation and some C16 at the boundaries. If she took the no competition model, as biochemistry should not work, she could not explain this, so then both the control and the patients would remain low. So clearly you need this competition, as biochemistry really works, to explain the patterns in the patients. But it also told us something about the severity of the disease. So here we had this no competition model, the artificial thing, and then we saw very little effect in both patients on the beta oxidation flux. But if we took the real model, then we saw differences between these two patients. Then one patient was predicted to have a small decline of flux and the other a very strong one. So this one in reality turned out to be the one with mild symptoms, and this one the one who died very early in life. So it was very much related to what we saw in reality. So yes, this is physiologically relevant for our patients, it seems. Although, of course, we have to go a long way to use this for treatment, but it's at least showing that we're on the right track. But the right track, of course, contains also the surrounding network. So I'd like to say a few things about that, how that would modify the system. So Marcel again started looking around this node in the network of coenzyme A. So he was collecting all the reactions that would directly influence this concentration as a consuming or a producing reaction. And I'll just pick out one of the simulations that he did, where he focused on a specific reaction called thioesterase. What this does is, if you have the palmitoyl-CoA or acyl-CoA, which is now in the mitochondria, it should go into the beta oxidation, but it can also be hydrolyzed. So then you just split this again in a fatty acid and a coenzyme A. So you take it out of the system. It can be recycled again. It costs energy to recycle it, but you can recycle it again to go back. But for the moment, you take it out of the system. And he modeled what was happening then. And he modeled both the wild type and the mutant situation. What you will appreciate here is that in the mutant situation, it didn't matter so much, just a little bit. But this is the disease case. And here you see the free coenzyme A, which is dropping much more drastically in our patients than in the wild type. You see the scale here. We start really dropping already at very low loading of the system. And you see that also the rescue by adding this enzyme, at least the rescue of our free coenzyme A, was much more prominent in this patient situation. So that's interesting. What then would it do to the flux? Well, he looked at the palmitoyl-CoA consumption rate. So this is how much substrate is being consumed. Well, it's no surprise that if you add an extra sink to this system that you start increasing the consumption. So that works. What was a surprise is that we also saw more going through this pathway. So here we, as a readout, we had the production of NADH, one of the products of this pathway. And what you see happening here is that even though part of there was an extra sink going in the other direction, nevertheless more was going through the main pathway, suggesting that this coenzyme A, which was released by this reaction, that is, was activating the whole thing, so that overall there would be going more to this branch. So I think this is really why you need modeling. This is, for me, a nice example of an emergent property. And this relates well to literature where people have overexpressed this enzyme and where they saw really higher oxidation of fatty acids by the mitochondria. So it fits. I have one final result, if that is okay in view of the time. This is another rescue that we found experimentally in this case. So here we had mice lacking this enzyme MCAT, and we put them to a stress test. So these mice are very, very good at fasting. They wouldn't die, they wouldn't have severe symptoms like the children. But if you give them an additional trigger, they get a more severe symptom. So here we had these mice after fasting for 4 to 6 hours at a very low temperature, 5 degrees. So they need a lot of energy to keep warm. So they need their fatty acid oxidation. And here you see, for instance, the blood glucose levels. And you see that they were normal before fasting. They were even not significantly different after fasting. But if you have after fasting plus cold, you see a significant difference. So now they start having a phenotype. You see they start accumulating a lot of acyl carnitines. Well, that's what we had seen before. Body temperature was a bit mixed. There were both in the wild type and in the mutant, there were some that could maintain their body temperature and others that could not. What we also saw was an increase in triglycerides. So these are the storage versions of our lipids. And this was more significant in younger mice. So these were 16 weeks old, so we would call them adults. These were 8 weeks old, we would call them young mice. You saw they were started accumulating fat in the liver, which is also what you sometimes see in patients to some extent, not severely, but to some extent. And Wengzhuang Zhang in the lab, she is a PhD student with my colleague Dirk-Jan Reijngoud, and she did lipidomics, and here you see a volcano plot, and you see a very prominent increase of especially these red dots, the triglycerides in the mutant. And if we then look at which triglycerides we saw, she saw often that there was in the acyl chains at least one C12 or C14. So she started quantifying that, and a triglyceride has 3 acyl chains, and then you see the first chain was always containing a shorter or medium chain length, so from C6 to C14, and even the second chain sometimes is C14, and then the normal nutritional like C16, C18 acyl chains, they were well, dominant only in the third chain. So there was a lot of medium chain and acyls in these triglycerides. So the way we view this now is that there is this problem in the medium chain conversion. There is a need, of course, to get rid of this accumulation of the metabolites, so it can be either directly incorporated into the triglycerides, but some of them are definitely longer than what we accumulate, so longer than the C6 to C10, so they're probably elongated. But altogether, it takes a lot away from this clogging of acyl-CoAs, and it releases the CoA. So we see this as an important mechanism for rescue, at least inspired by our model. This is how we interpret this, not necessarily as a problem in itself, like you would think, triglycerides, I don't like to see them in my liver, but it's much better, of course, than a shortage of coenzyme A. So this is some first glimpse on how the surrounding networks affect our phenotype. So what are our challenges now? We have a big project starting, and for those of you still looking for a PhD, there are also positions. So I'm coordinating a training network in Europe, and I'm also participating in another one. So one is called Polymer, that's the one I'm coordinating. The other is Perico, where I'm a partner. And what we are focusing on is, well, first of all, we need to get more of this surrounding network in our models, but also we need to develop analytical methods to see all these things, to make interventions in it. But the key thing at this moment, I think, in systems medicine, if we want to bridge between the molecular level, what's happening in the cell, and what's happening at the whole body level, we must have in vitro system in the test tube where we can culture tissues that resemble real tissues, not like immortalized cell lines that are typically cancer cell lines, but really something that resembles a liver tissue, and preferably also a patient liver tissue. So we work with induced pluripotent stem cells derived from patients, and we're trying to develop these into mini livers that can be analyzed to generate the data for the models. So that's how I see this developing in the coming years. And with that, I'd like to close. I have mentioned a number of people along the way, but I did not mention the key person in the lab, Albert Gedding. He's our technician, and our very good collaborator, clinician, Terry Derricks. He is a key figure in treating patients with these disorders and organizing the research throughout Europe. And our clinical chemist, Rebecca, and Dirk Jan, who is my colleague for many, many things, who is also here in the audience, I just noticed. So with that, I hope this was of interest for you.
Yeah, thank you so much, Dr. Bakker, for this incredible presentation. It's very interesting how you sort of harness systems biology to improve treatment. I think first, we'd like to sort of have an interactive discussion, but anyone have any questions on Dr. Bakker's presentation? We can just sort of speak up or add it to the chat.
I could stop sharing the slides unless you would like to see them back.
Oh, yeah, I think, it would be best if you stop sharing the slides.
No, sir, no, that's not pause share. That's what I want. Yeah. So, yeah, looks like I can see you full screen now. Okay.
Yeah.
Hi.
Yeah, so I had sort of, I had one question. So I was curious because I know you're coming at it from more of a biochemical perspective. So one of the things you mentioned in this presentation was how modeling competition is sort of crucial to have predictive models. Can you think of any other examples of sort of issues that modelers maybe aren't focusing on enough that really should be added into metabolic networks, things like that?
Other aspects that should be added. Well, as you see, I'm very much a kinetic modeler. You see a lot of models that are, for instance, the genome scale models, they do not contain any kinetics at all. We also see the whole body models, which I think are extremely important to see, how do the different tissues connect, so just to get the complete physiology of the person. But then you often see linear kinetics, like a reaction is just a constant times the concentration. And I think a lot of the interesting properties, especially something like bistability or the breakdown of a system when you overload it, these are emergent properties that often come from these kinetics, and we somehow have to bring these worlds together. I think that's really a challenge. You see, I come from biochemistry, I've done it this way, but we are also pioneering the other ways. This is really a challenge because I'm aware we cannot do this for the whole body. This will take ages and ages. So for instance, what we are pioneering now is to take a whole body model with simple linear kinetics and see what would happen if we would put our model in that context, to see if something like the bistability is still maintained and how certain metabolites, for instance, that are produced in the gut and that may have an effect on this, how, if we can really link this to the whole gut metabolism in a simplified way, but still get the right interactions into this very specific model. So that's a way I can see this starts to develop. But I definitely believe that this example is one out of many, that there is importance for considering the details, but you have to be very, very critical when you do it, because you see how much work it is for such a tiny piece of our system.
Thank you.
Any other questions? I see many people nodding.
We have a question from James, just whether or not your biochemical models are deposited in like a public repository like biomodels, or if they will.
Yes, they are. Yes, they are. Yeah. Yeah, we also work together a lot with Jackie Snoop. So he will be the speaker in 2 months. So he has developed this JWS repository. So of course we also get to biomodels from there on, because they connect. The nice thing, I like to make a little advertisement already for Jackie, who is speaking in 2 months. The nice thing about his work is that they also do quite some curation of the model. They've done that from the beginning to really show that they can reproduce basic characteristics as they were published, and you can also do modeling. If you're not a modeler, you can just happens. But he will tell more about that. But all our models are reposited there. Yeah. Who else?
Well, I had an additional question about something you mentioned at the end. You said like the real way to pioneer further is to actually work with in vitro models. So I'm curious to know, like, which community do you think it's important to reach out to work together on this project?
To work together on this project. Whether I think this is important?
No,
I cannot hear you. I cannot hear you very well. Sorry.
Oh, sorry. Like, which scientific community do you think we should reach out to sort of develop in vitro models to analyze from a systems perspective?
Yeah. So at the moment there is a huge development going on in these in vitro systems, also for personalized medicine. So people starting from iPS cells, so stem cells, which you can generate, for instance, from skin cells. So you have a skin biopsy, but also from blood cells, from urine even, you can isolate stem cells. So you can have them from the patients, and you can re-differentiate them into a liver or a brain. And there are huge developments there at the moment, even making coupled mini organs, like different organs coupled to each other, so that you get an in vitro mini human. And there I think you can get loads of interesting parameters. I've seen already, I think it was Ben Moaz from Israel, he was doing labeling studies with stable isotopes following the metabolism in such a model system. And I think that is the future if we want to bridge between the human and what's happening molecularly at the cells, and to model that properly. Yeah. So there is a huge community out there. It's not necessarily a modeling community at the moment. It's also a bioinformatics community a lot. I mean, doing proteomics, transcriptomics, and analyzing the data, more like pattern recognition. There's loads of things happening there. Microscopy is, of course, the first thing that you can do with such systems. So there is some development to contribute to. Yeah.
We have a question from, I think we're pronouncing this right, Chabaro, but he says, I was wondering what you think is the biological relevance of reduced beta oxidation flux at high palmitoyl-CoA concentrations.
Yeah, so what's the biological relevance of this? Well, if you get into a situation of fasting, but also a situation of cold, which is also a trigger which we should should be able to cope with as organisms, then the natural response of the body is to liberate lipids from the adipose tissue, so from our fat mass. So then we get a much higher loading of the liver. And normally, of course, we should be in this healthy situation where the liver can still work with this. And as we saw already, I mean, our model is a bit simple in the sense that it's the naked beta oxidation, and surrounding pathways are probably helping it to deal with this overload. But in a patient situation, this sensitivity for overload is likely to become more. We saw this in our model. And then, of course, it may be a serious cause for breakdown. But we're also thinking of the obese situation. I have, for instance, a postdoc, the same mathematician postdoc who worked on this Bistability, which I showed. His project is on fatty liver, where the liver starts accumulating a lot of lipids, where apparently the beta oxidation is not working properly. And the question is whether this Bistability may help us to understand why you can get people from, let's say, a disease state back to a healthy state, or vice versa. So if there are interventions that you could make that get people again on the right branch, or maybe, let's say, it could even be daily interventions, like what does a morning walk do to this system? I mean, a morning walk would be utilizing the NADH, so it could be directly linking to our model. But also, what do the gut, in our gut, our microbes are producing a lot of metabolites that have an effect, like the short-chain fatty acids. We're working on that. So we're trying to get a handle on how this Bistability could really work in vivo, and whether we can use it to optimize therapies. Is that an answer to the question?
Sounds like a good answer to the question. I just have a question about what you think about combining several types of modeling with, do you think it would add value to what you've already discovered if you added, for example, spatial modeling of what's going on within a cell, for example? Or do you not think that is necessary? Do you think enzyme kinetic models are sufficient?
No, I think it's extremely important, especially in pathways like this one. It's extremely important. I started my career in modeling of glycolysis, and then you have very abundant enzymes and metabolites. When they start diffusing, they rapidly see, just by simple diffusion, assuming this is a mixed vessel, you can very well simulate what's happening. But here we have metabolites that are very sticky, like the lipids. Also the enzymes tend to form clusters, and the degree to which this happens may differ, but this will certainly have an impact. And it may also improve the situation. In fact, if there is a lot of channeling of metabolites, it may avoid the clogging. And this is definitely something we need to investigate further. Yeah. For instance, one of the things that you see if you look at the binding of enzymes to each other is that some of these enzymes in our pathway, they bind to enzymes in the respiratory chain that consume the NADH that is produced. So, and that will probably be a relief to the system, and also maybe something that is different between different people. Because if just the binding between two enzymes is defective, it may not give an immediate problem if it's a little bit defective, but in combination with a mutation, you might imagine this is the case. So I'm kind of speculating, but I think the nice thing of modeling is that you can rationalize your speculations to see at least whether they are feasible. And if then they are feasible, then you also, of course, need experiments to test them, but you can also do your experimental design then based on your model. So yes, I believe this is extremely important. And also, I mean, as I said already, I like to think and explore this linking of whole body models, physiological models, to these detailed biochemical models, because they both have their strengths and their weaknesses. And if you can link them, you might get the best of both worlds.
Thank you. Do you have any other questions? Sorry, Joseph, I cut you off a bit.
Yeah, Dr. Bakker, like, thank you so much, you know, for bringing the seminar. And like, it's fascinating to hear from someone with, like, you know, a rigorous biochemical background. It's, you know, adds in sort of a lot of like what the overall strategy for modeling should be. So thank you so much for this presentation.
Well, thank you for being here. It was really exciting to do it this way. And I apologize if this was too much biochemistry, but this is my way of thinking, and also I think a field where the modeling can help a lot. So I hope I could bring across that message, despite some of the details of the biochemistry.
Yeah. Well, thank you, and you know, everyone, please join us the first Tuesday of March for our next monthly seminar.
Okay. Goodbye, everybody.